Hello, everyone. I hope I am audible very loudly and clearly. So, here it is, the most awaited live streaming IITV x MIT VPU. First of all, congratulations to all the participants of BioQuest. A huge shout out to all the students who took part in the BioQuest examination, and a special congratulations to those who have qualified for the round 2. Today, we welcome you all to this live stream where I am your host, Sanvi TS from IITB, joined by Mitrayu, uh, Shreya, and Angel. Tonight, we are bringing you a crash course on tomorrow's exams topics, along with a quick concept reviews, sample practice questions, live Q&A, and interactive problem-solving sessions. So grab your books, notes, snacks, and maybe your dinner, and get comfortable because it's going to be one exciting ride. Let's get it started. Over to you, Angel. Angel, over to you.
Hey everyone, so Angel this side. I am a student from IIT Bombay and I'll be explaining the first three chapters to you. So, the first slide, as you can see here, is understanding the tests for biomolecules and the key identification methods. One of the first One of the first, sorry for the delay. One of the first tests that we'll be going through is the iodine solution test. So, just to tell you guys a little bit about what iodine solution is, it's a mixture of I2, which is free iodine, and potassium iodide. They are both not soluble in water, so through some other chemicals, they are made soluble in water. Now, what happens is the iodine solution reacts with the amylase and the amylopectin in the starch. The amylase, when reacted with the iodine solution, turns blue black in color. And the amylopectin, when reacted with the iodine solution, it causes a helical structure to form. So, in turn, when the iodine solution reacts with the starch, the blue-black color is observed. Hence, if a starch, when added to iodine solution, turns blue-black color, it indicates that the test is positive. Secondly, let's talk about the Benedict solution test. So, Benedict solution test is for reducing sugars and changes color upon heating. Now, can you all let me know, if anyone does, what reducing sugars are and try to give a guess on some examples of reducing sugars? We'll wait for a bit to see your answers. To repeat the question, could you go back to the previous slide? Yeah. To repeat the question, I'd like answers from you guys on what reducing sugars are and some examples of reducing sugars. So I'll give you a hint. Glucose is one of them. All right. So fructose is indeed correct. Sucrose is also correct. So those are very correct answers. So essentially, reducing sugars are a group of sugars which have free aldehyde and ketones. So essentially, what this reaction is, it's a redox reaction. The Benedict solution consists of many other compounds, but majorly it's a redox reaction which causes the colors of the sugar to change when added on it. In the next slide, I will let you know more about it. Now, the third test is the biorate reagent, which detects proteins. So, biorate not only detects proteins, it also detects polypeptides. And basically, the higher the purple color, as written here on the biorate reagent, when added to proteins, it turns purple color. So, the purple color depends on the number of peptides in the protein. So, the higher the protein or the longer the protein chain, the more purple color will be observed. Now let's talk about the ethanol test, which helps in identifying fats. So a lot of you guys may have studied the CO2 test, in which the milky white cloudy emulsion appears. So similarly, when ethanol is added to fats, a milky white cloudy emulsion appears in the sample, and so this is one of the key tests to fat. What you guys have to remember is the change in color. That is very important, and it helps and it gives you hints on what the compound can really be. The last test is the DCPIP test, which turns colorless when added to a sample containing vitamin C. Do you guys know what vitamin C is? I mean, what is the real name of vitamin C? I'll wait for your answers in the chats for 2 minutes. To repeat the question, do you guys know the actual scientific name of vitamin C? If you do know, then you're requested to write it down on the chats. We're waiting for you. Not where vitamin C is found, but the real name of vitamin C. It's an acid, that's a hint. That is indeed correct. So, vitamin C is known as ascorbic acid. So, yeah, the key takeaway is that DCPIP test turns colorless when added to vitamin C. Let's proceed to the next slide. So, as you guys can see here, it's a summary of the important things that you're supposed to remember for your test as well as for later when you guys will be studying biology. So, like we discussed, iodine test for starch. The color of iodine is orange brown, and when it's added to starch, the starch turns into a blue black color if starch is present. But if it's not, then there is no change in color. So let's remember iodine solution is orange brown, which turns to blue black on a positive test. Similarly, for Benedict solution, the color of the reagent is light blue. And when it is added to appropriate sugar, as mentioned, reducing sugar, it will turn green to brick red. That is the spectrum of the color. So that means that the key takeaway is that light blue changes to brick red or green for sugars present while Benedict solution test happens. The third test is ethanol for lipids and fats. The color of ethanol is colorless. And if the test is positive, that means if lipid is present, then it's a cloudy emulsion. If not, then the solution remains colorless. So, what remains colorless? The ethanol solution. Next and the last test is the biorate test for proteins. So if the color of biorate is blue, the biorate solution, and if protein is present, it turns to lilac purple. And if it's not present, then it remains blue. I would request you all to take a screenshot of this. This would be really helpful for you guys when solving questions in the future. Let's move on to the next slide. I hope you all have taken screenshots. So let's now study a little bit about what enzymes are. So enzymes are biological catalysts that speed up chemical reactions in living organisms. They are highly specific in nature and follow a model which is hypothesized to be lock and key model. Moreover, their activity is affected by temperature and pH. So, as you guys can see in the diagram, the enzyme sort of has these troughs, which are like where the substrate binds. And because they are highly specific, so each enzyme's trough is in such a way that only a specific substrate can bind to it to form an enzyme substrate complex. So, the lock and key model is sort of unique for every enzyme. That is, the thruff is unique for every enzyme. What essentially happens in enzymes is actually how they speed up the chemical reactions. It's by lowering the activation energy needed. If you all are really curious on what activation energy is, you all can comment in the live chat and we'll help you through it. And if you guys are really interested, then we'll send you some details about it as well. Let's move on to the next slide. So, how exactly did scientists know that enzymes speed up chemical reactions? They did a very interesting test, which is an experiment to measure enzyme activity using hydrogen peroxide and potato. The test goes as follows: we add hydrogen peroxide to a test tube, then add fresh potato strips, and then we see that bubbles are released. Now, how do we know that it is oxygen? We do the glowing splint test, which relights, which implies that oxygen is present. Now, if I do the same test, that is hydrogen peroxide, potato strips, and add manganese dioxide to it, then the bubbles are faster released. So this means that manganese dioxide is an enzyme because the reaction is happening faster. Now let us take a look at some more interesting facts about this. Basically, if we add boiled potatoes instead of fresh potato strips, then bubbles are not formed. And if we don't add H2O2, then there is no reaction. And if it's a cold environment, then the bubbles are slow. All of this leads to the conclusion that enzymes work best at the right temperature and pH, and only if the right substrate is present. As we can see, we needed hydrogen peroxide, fresh potatoes, and a sort of balanced environment for bubbles to release. And with manganese dioxide addition, the bubbles released faster. However, when we did not satisfy any of these conditions, then the reaction was not up to mark. So, enzymes are specific and work at the right temperature and pH only if the right substrate is present. I hope this is clear to everybody. If not, you all can use the comment or the chat box to ask your questions. Let's move on to the next concept. So, there's a question for you, everybody. Let's try answer this as quick as possible. So the question is as follows. A student conducts an enzyme experiment and finds that at 60 degrees, the enzyme activity drops drastically, even though the substrate is still present. What most likely caused the loss of activity? The option A is the enzyme denatured at high temperature. Option B is the substrate evaporated. Option C, the product became an inhibitor. And option D, active site became larger. Let's wait for 2 minutes while you guys cook up the answers and type them out in the chat box. So we're getting a lot of answers. Let's see if it's the right one. So the correct option is A indeed. The enzyme denatured at a high temperature. Can we move back to the question? So just to explain in gist what the question means is that as we studied before, that enzyme activity depends on temperature. And here at 60 degrees, the activity drops drastically. Out of all the options, let's say the last option, if the active site became larger, then it's not necessary that it would have caused a loss of activity. Similarly, with option C, the product became an inhibitor. And for option B, the substrate evaporated. That is a rather silly option, right? So, as for option A, the enzyme denatured at a high temperature is possibly likely because 60 degrees is a really high temperature for body enzymes. And so, denaturing would cause loss of activity. Let's move on to the next concept. So, the final concept that I'll be covering is the circulation system in organisms. By the way, if you all still don't understand the solution to the previous question, you guys can type it out in the chat box and we'll help you there. So let's talk about the single and double circulation in organisms. So single circulation involves blood flowing through the heart once per cycle. It is usually found in fish for maximizing oxygen efficiency. Whereas double circulation allows blood to be pumped to the lungs and body separately, which means that the blood flows through the heart twice as times. And double circulation is seen in mammals, which ensures effective oxygen transport. Let's look at this concept a little more in detail. So, for mammals, for example, why is it that we need more effective oxygen transport? We have such a huge body, and our major goal is, of course, maximizing oxygen efficiency, but also thinking that all that oxygen reaches to all parts of our body. You guys can think of it this way. For fishes which have single circulation, the fish's heart pumps dirty deoxygenated blood to the gills. In the gills, the blood picks up oxygen and becomes clean. This clean blood then flows directly from the gills to all parts of the body. After giving oxygen to the body, the dirty blood comes back to the heart. So essentially, the blood only comes to the heart once to get oxygenated. Whereas for double circulation, it's like a two-story house where water is pumped to the first floor, then pumped again to the second floor for better pressure before draining back to the pump. So I hope you all understand the concept of single and double circulation systems in organisms. Let's move on to the next concept. So this is where you all can understand the double circulation a little bit better. As you can see here, this is a very detailed picture of the heart, which you guys should be remembering, at least the labels of the parts. So I'll briefly go through the hard parts, but it is very much advisable that all of you guys read them in a little more detail so as to understand their functions better. So the vena cava, which is also known as the inferior and superior vena cava, is on the top part of the heart. It is where the deoxygenated blood from body parts enter into the heart for oxygenation. It enters into the right atrium, as you guys can see from the arrows. So basically, our heart is divided into 4 chambers, and it enters the first chamber known as the right atrium. So as written here, it receives deoxygenated blood from vena cava and pours into the right ventricle. After this, a valve, which basically is used to prevent backflow so that the blood only flows in one direction. This valve between the right atrium and the right ventricle is known as the tricuspid valve, and as written here, it prevents the backflow into the blood. After this, it enters the right ventricle, where it is pumped, and the function of the right ventricle is pumping deoxygenated blood into the lungs through the pulmonary artery as shown. So after the right ventricle, it enters the pulmonary artery and then gets sent to the lungs, where it is essentially oxygenated. And after that, the blood, which is oxygenated, enters back into the pulmonary artery from the lungs to the heart. The pulmonary vein then carries the oxygenated blood from the lungs to the heart. After that, the blood enters the heart in the left atrium. So as written, it receives oxygenated blood from the pulmonary vein and pours it into the left ventricle. Similarly, a valve is present there too. Finally, the aorta carries oxygenated blood from the heart to all the body parts, and it generates immense pressure since we need to supply blood all the way from our brain to our little toes. Two more parts of the heart are the semilunar valves and the septum. The semilunar valves, as its name suggests, prevents backward flow of blood once the blood is pumped out of the heart. So it is present near the aortas because the blood is pumped to the body from the aortas. And the septum is the muscle wall which divides the heart into the right and left sides. So it prevents the mixing of the oxygenated and deoxygenated blood. So I have a fun question for you guys. If you all know, veins usually carry deoxygenated blood, but over here the pulmonary vein seems to be carrying oxygenated blood. Do you guys have a possible answer to this? We'll wait for your answers while my course take on. All right, guys, one more question. So, why does blood pressure drop significantly in veins compared to arteries? We'll wait for y'all's answer. Let's do this real quick. So, I'll repeat the question for y'all. Why does blood pressure drop significantly in veins compared to the arteries? The options are as follows. Veins have stronger muscle layers. That is option A. Option B is resistant to flow is highest in capillaries before veins. Option C is arteries are connected directly to the heart. And option D is veins absorb nutrients and water. So it seems like there is an answer war in the live chat. Let's have a look at the right answer. Can we have a look at the right answer, please? So the right answer is B indeed. Let's have a look at the question. So, as stated, the option B says resistant to flow is highest in capillaries before veins. So, capillaries are very, very thin, and veins are slightly larger. Capillaries are used to provide blood into the muscles and to each and every cell, right? So, they are very, very small, hence, offer large resistance. And that is why blood pressure drops significantly in veins compared to arteries.
Thank you so much, Angel. So, from here onwards, my friend Mithrayo will be joining us.
Hi guys, Mitrayo here from MIT WPU Pune. I hope I am audible enough. Okay, so I'll be starting with transport in plants. So we all know all organisms need to obtain substances from their environment, and for plants, these substances are carbon dioxide and water for photosynthesis, and mineral ions which they absorb from the ground. And hence, like our circulatory system, plants also have a circulatory system. And it composes of two main structures, which is the xylem and the phloem. So xylem is a plant tissue made up of dead empty cells joined end to end, and it transports water and mineral ions and helps to support the plant. Phloem, on the other hand, is a plant tissue made up of living cells joined end to end, and it transports substances made by the plant, such as sucrose and amino acids. Um Xylem has Xylem is lined with dead cells called lignin. Lignin is a hard, strong, waterproof substance that forms the walls of xylem vessels. This allows a continuous movement of water throughout the plant. Now, in plants, there is one important concept about transpiration. So, transpiration can be defined as the loss of water vapor from a plant. So, we may remember there are openings on the surface of the leaf called stomata, and these are more on the underside of the leaf in the lower epidermis. So the mesophyll cells inside the leaf are each covered with a thin film of moisture, and some of them have air spaces in them. So the water vapor diffuses out of the leaf through the stomata, passing through these cells. Water from xylem vessels in the leaf then travels to the mesophyll cells by osmosis to replace this. This creates a transpirational pull which helps draw more water up the xylem. So, like any other life process, there are multiple factors that affect the rate of transpiration. So, the higher the temperature, the greater the kinetic energy of water molecules. This means that water evaporates faster from the surface of the mesophyll cells, and the water diffuses out of the leaf more into the air and more faster. On the other hand, on a windy day, for example, the air around the leaf, which contains a lot of water vapor that has just diffused out of the leaf, is quickly moved away. This means that there will always be a diffusion gradient for the water vapor because there is less of it outside the leaf than in the air spaces inside the leaf. So, water vapor diffuses out of the leaf faster on a windy day than on a day when the air is still. Hence, a strong wind increases water loss via transpiration. Transpiration rate is also affected by humidity. Humidity means the moisture content of the air. The higher the humidity, the less water vapor will diffuse out from the leaves. This is because there is not much of a diffusion gradient for the water between the air spaces inside of the leaf and the wet air outside it. Hence transpiration decreases as humidity increases. Moving on to wilting. Uh, Wilting happens when water loss exceeds water uptake. Cells become flaccid and tissues lose support, and the plant bends or collapses visibly. Now, in order to measure transpiration, we use an instrument known as the photometer. A photometer has, in simpler terms, we use photometer by performing the following steps. We first cut a leafy shoot under the water, insert it into the photometer, and while cutting, also we ensure it is a slant cut. And once we have submerged it into the photometer, we take it out of the water and make it stand it on the bench. And post that, we check that our photometer is airtight, and that there are no air bubbles in it. We then trap air bubbles in a capillary tube and then measure the distance moved by the air bubble over time. The rate of respiration will equal to the bubble movement speed over a particular time period. Guys, any doubts till now? Okay, so we have a small question for you. Two identical sunflower plants are grown, one in saline soil and one in pure water. Despite equal watering, the saline grown plant wilts daily. Which factor explains this best? Stomatal closure due to salt, root pressure increase in saline condition, lower soil water potential reduces water uptake, or D, xylem blockage by saline particles. Guys, please post in your answers in the comment section. Okay, so I can see there is quite a competition between C and D. So let's check the correct answer now. It's C, lower soil water potential reduces water uptake. Okay. Now moving on to the next concept that I will be taking over is understanding excretion and kidney function in homeostasis. Okay, so blood, when we talk about excretion, it refers to the removal of toxic substances and anything in excess from our body. And one of the most and the major organ that is involved in this is kidney. So one of its major functions is filtration. So blood enters this kidney and waste is filtered out, and it is and the urine is then stored into the bladder, which is then removed through urethra. So our kidney consists of these small, tiny functional units called nephrons, which facilitate the filtration and reabsorption processes. Glucose and useful salts are reabsorbed, while any substance in excess and excess of water which is not required by our body is excreted out in the form of urine. And thus, by excretion, the body maintains an internal balance, which is homeostasis. Homeostasis here refers to the maintenance of constant internal environment. Okay, so moving on to our next concept, which revolves around urinary system. We have four major key components of a urinary system. The first one is obviously our kidney, which are bean-shaped organs that filter out the blood. The major filtering unit in our kidney are the nephrons. Followed by this, we have the ureter, which is a tube that connects the kidney to the bladder. After filtration, the waste products that are produced, which eventually becomes the urine, is transferred from the kidney to the urinary bladder via the by the ureter. Now, this urine is then stored in the urinary bladder. This urine has excess of water, salts, and urea. Urea is formed. By any chance, you all know how is urea formed in our body? You all can use the comment section to comment your answers. Okay, so excess of amino acids that cannot be stored by our body are converted to urea, and this urea is then excreted out through our urine. Now moving on to urethra, it is a tube that connects the bladder to the exterior where urine is released. Okay, moving on to the last concept that I will be covering is human nutrition, which is digestion. So we all know that there are five major steps involved in digestion. It starts with ingestion and ends with assimilation. So ingestion of food occurs in the mouth where it is chewed and mixed with saliva. So here two major processes takes place. which is the mechanical digestion and chemical digestion by salivary amylase. Following this, uh, following digestion, following this, digestion takes place, followed by absorption, which is the movement of small nutrient molecules and mineral ions through the walls of intestines and into the blood. Following absorption, the nutrients are then absorbed by our human body, and finally, the waste products of digestion are excreted out through the process of digestion. So now, the major organs involved is the stomach, where acid gastric juices break down food, preparing it further for digestion. Nutrient absorption, on the other hand, primarily takes place in the small intestine, and which is facilitated by villi that increases surface area. The large intestine absorbs most of the water and consolidates waste before it is excreted out from the kidney. So, food is ingested using the teeth, lips, and the tongue. The teeth then bites or grinds the food into smaller pieces. This increases the surface area. This begins the process of physical digestion. So there are two types of digestion that take place in our body. The first one is physical digestion, where there is breakdown of food mechanically, but there is no change in the chemical structure of the molecules in the food. On the other hand, chemical digestion refers to the breakdown of large molecules in the food into smaller molecules so that they can be absorbed. So a point to be noted is in mouth, both physical and chemical digestion takes place. Physical digestion is taking place by teeth, while chemical digestion takes place by salivary amylase. Salivary amylase is an enzyme that is secreted by salivary glands, and it allows partial digestion of starchy foods, and it is converted, and the end product is maltose. Following this, your food moves down through the esophagus into our stomach. So the stomach has strong muscular walls, and these muscles contract and relax to mix the food with enzymes and mucus. Like all parts of the alimentary canal, the stomach wall contains goblet cells which secrete mucus. It also contains other cells which produce enzymes and others which make hydrochloric acid. One of the key function of hydrochloric acid is it kills or it kills any harmful organisms that might cause an infection. The enzymes produced in the stomach digest proteins, so they are called proteases. So proteases digest proteins and convert them into amino acids. As a result of hydrochloric acid being produced in the stomach, the pH is very low in the stomach. So the pH is about 2 in the stomach, and this pH is also optimum for the protease enzymes to act in the stomach. So following this, the stomach can also store food for a quite long time. And after 1 or 2 hours, the sphincters at the bottom of the stomach open, and the less partly digested food now moves into the duodenum, which is the first part of the small intestine. So the complete digestion takes place in the small intestine. The small intestine is a part of the alimentary canal between the stomach and the colon. And now, here major processes that take place is first, pancreas through the pancreatic duct secrete the pancreatic juice, and finally major digestion takes place here. And apart from that, absorption of nutrients and most water also takes place in small intestine. The pancreatic juice secreted contains the enzymes amylase, protease, lipase, which converts again starch to maltose, proteins to amino acids, and the fats to glycerol and fatty acids. The specific part is the ileum, where small intestine digests stuff. Pancreas and livers are both important organs in the digestive system, but they are not the part of the alimentary canal. Foods do not pass through them. We have seen that pancreas secrete a fluid containing enzymes which help with the digestion of food, even in the duodenum, which is the part of the large intestine. The liver also secretes a fluid that helps with the digestion, which is called bile. The bile is made by the liver and stored into the gallbladder and is then secreted through the bile duct, which is then mixed with the food in the duodenum. Bile is a yellowish green alkaline watery liquid which helps to neutralize the acidic pH as well. Next slide, please. So again, a question for you all. A patient has a medical condition where bile secretion is blocked. Doctors observe reduced fat digestion and incomplete protein digestion in the small intestine. What is the best explanation for these observations? Stomach enzymes are overactive, pepsin and trypsin are both denatured. Bile cannot neutralize stomach acid, affecting trypsin activity. Amylase is failing due to temperature changes. Please put the correct answer in the comment section. Okay, so the correct answer is C. Bile cannot neutralize stomach acid, affecting trypsin activity. So, bile, as mentioned earlier, is secreted by the liver and stored into the gallbladder. Bile helps in neutralizing the acidic pH when food comes from the stomach. And hence, if bile doesn't function well, it cannot neutralize stomach acid. And trypsin is a specific enzyme which only operates at a higher pH. Hence, trypsin activity is affected, which leads to incomplete protein digestion.
Hello, guys. Thank you so much, Mitrayu. So, from here onwards, I will be taking over. So, first, let's understand the photosynthesis. That is quite a common topic we all have understood since quite a young age, right? Since almost like third standard, we must be understanding plants and photosynthesis. So, let's go through the basic concepts and some uses that you guys use in your daily concepts. So photosynthesis equation is 6CO2 plus 6H2O and it gives you this compound. I'm pretty sure everyone knows what's the name of this compound. Maybe we all can understand which compound this is. Type it down in the chat. What is this compound's name? What is this compound's name? Okay, I'm getting a lot of answers out here. Glucose. Correct. So glucose serves as an energy for the plants and it is stored in the starch. Oxygen is a byproduct and it is essential for the aerobic respiration in all the living organisms of Earth. Plant utilizes these glucose for growth, reproduction, and cellular processes. So let's understand the limiting factors of photosynthesis and the stomatal function. Light intensity affects the rate of the photosynthesis significantly. Carbon dioxide concentration is crucial for the plant growth. Temperature influences the enzyme activity in the photosynthetic processes. Stomatal opening regulate glass exchange and water loss. So here is a brief explanation of the structure of a leaf. How does a leaf look? Have you ever examined in front of your eye? But what about how it looks in a microscopic view? So this is a bisection of a leaf in a microscopic view. You can see here that the sugar exits the leaf through the phloem, the water and the minerals enter the leaf through the xylem, and there is a bundle of a sheath cell. This is called as a leaf vein. Out here you can see the stoma and the guard cells. So since you all are 9th and 10th standard kids, you must have got an exercise from your biology teachers to draw the stomata and to show it closed and open. So this is how you exactly draw, right? The guard cells and the stoma. So the oxygen and the H2O exit the leaf through the stoma. Whereas there is a nucleus, there's a guard cell, and CO2 enters the leaf through the stoma. There is chloroplast and stoma. And you can see all the other minute details of your leaf, which you usually don't have it in your biology textbooks. But here it is: trichomes, cuticle, upper epidermis, parasylade, mesophyll, air spaces among the spongy mesophyll, lower epidermis, cuticle. You can also see a leaf cell. So you must have also got an exercise from your biology teachers about the leaf cells and the animal cell, right? What is the difference between a leaf cell and an animal cell? Comment down on the chat. I repeat, what is the difference between an animal cell and a leaf cell? Okay, I am seeing some answers out here. So, the difference between an animal cell and a leaf cell is the animal cell lacks a cell wall. That is right, Rajkumar Gupta. Good job. So, here is a question for all of you guys. In an experiment, increasing light intensity no longer increases the rate of photosynthesis, even though chloroplasts are functioning well. Which factor is most likely limiting the process now? Option A: temperature is too high. Option B: carbon dioxide concentration is too low. Option C: starch levels in leaves are high. Option D: chlorophyll is saturated. Okay, I'm seeing a lot of answers out here. And there's a huge competition between B option and D option. Carbon dioxide concentration is too low or chlorophyll is saturated. Let's see what's the correct answer. The correct answer is option B. Carbon dioxide concentration is too low. That is correct, guys. So let's go to understand the biological hierarchy and the binomial nomenclature. Taxonomy of an organizes the living things into categories based on similarities. So you guys must be having a chapter on the categories of the different classifications of animals and plants. So in the same way, here we are going to categorize it on the same similarities. The binomial nomenclature gives each species a unique two-part name. Do you guys know what is the name of the humans that is given scientifically? The unique two-part name for humans? Comment down on the comment section. I repeat the question. What is the scientific name given to the humans? The two-part name. Great. I can see a lot of smart kids in the live chat. So there are many answers out here. Yes, the correct answer is Homo sapiens. Wonderful, guys. So the arthropods are characterized by the exoskeletons, segmented bodies, and jointed limbs. Domains are the highest classification, including archaea, bacteria, and eukarya. Animals are classified as vertebrates. Vertebrates are those which have a backbone. Invertebrates. Invertebrates are those which do not have a backbone. And viruses. This is a new different thing. They are unique acellular entities that require host cells for replication. I'm pretty sure everyone knows about coronavirus, right? So here is the classification of the plants, and here is a classification of the vertebrates. So in your ninth and 10th, you must have gone through this, a monocot and a dicot. And also in the round one examination, you had come across this kind of question, right? A monocot and a dicot. So remember this diagram very carefully. A monocot diagram looks like this. The seed, the root, the vascula, the leaf, and the flower has three petals. Whereas a dicot has two cotyledon, they are tap roots, the vascular are ringed, the net veins, and 4 to 5 petals are there. Remember this because this might come. And as for the classification of vertebrates, mammals, they have fur on the skin and everything that you guys have basically. For example of a mammal is horse, dog, and ourselves. Birds, they have 2 legs, 2 wings, of course you guys know. Examples are eagle and parrot. There are reptiles, there are amphibians, and there are fishes. So you guys know all about the vertebrates and the plants now. So from here onwards, my friend Shreya will be taking over.
Thank you, Sanvi. Hello, everyone. I'm Shreya, and I hope you're enjoying the session so far. We have discussed several biological processes till now, and if we mix technology with it, what do we get? Biotechnology, and it is one of the most important and increasingly essential and relevant industries in today's world. Biotechnology is essential in several industries like bread production, which utilizes yeast fermentation, which enhances flavor and texture. Biofuels are produced from renewable resources, providing sustainable energy solutions and reducing fossil fuel dependence. Enzymes play a crucial role in various biotech processes, improving efficiency in food production and pharmaceuticals. Now, many of us are lactose intolerant. So biotechnology is coming up with a solution for that too. Lactase production helps in dairy digestion, making lactose-free products accessible for those with lactose intolerance. Yeah. And we move ahead. Fermenters like pause enhance microbial growth and production efficiency. Penicillin, we all know about penicillin, right? Penicillin production utilizes fungi to create antibiotics for bacterial infections. Bioprocessing allows for large-scale production of essential biological products. Understanding fermentation processes is key to improving yield and effectiveness. So, time for a new question. In a fermenter producing penicillin, sterile air and controlled temperature are constantly maintained. Why is this important? So, you have a few minutes. Go ahead and shoot your answers. Yes, I think we have a clear winner with us. So, our answer is option A, to prevent contamination and optimize microorganism growth. It is important. These conditions are very important for avoiding contamination because even a slight bit of contamination can result in disastrous results. We can move forward now. So, genetic engineering. What do you all know about genetic engineering? I'm sure you must have watched several movies like Jurassic Park and The Amazing Spider Man. So, what do you think it involves? The entire movies are based on genetic engineering, whereas whether it is cloning in Jurassic Park to bring back our long lost dinosaur friends, or it is gene editing by introducing spider genes in the amazing spider that is Spider Man, that is Peter Parker, and that is how we get our eternal savior. So, the key steps in making insulin using genetic engineering are: identify the insulin gene from human DNA for cloning, insert the gene into a plasmid vector for transformation, introduce the recombinant plasmid into bacterial cells to express insulin, harvest and purify the produced insulin for medical use. Now, I think that you all must be a bit confused as to what is going on. So, we have a better explanation for you in the next slide. So, plasmid vector. Plasmid is basically an extra chromosomal genetic material that are present in several bacteria and microorganisms. This plasmid vector, shown in the figure, is cut with restriction enzyme. This is basically the scissors in the world of microbiology. The gene to be inserted is placed in the cutout portion of the plasmid vector. These gene, the inserted gene and the plasmid vectors have sticky ends which helps in the binding, and it results with the help of DNA ligase in a recombinant plasmid that contains the gene which we want to be present in the plasmid. The recombinant plasmid is then inserted back into the bacterial cell, and this cell now shows the results of the recombinant plasmid by showing the effects of the gene that has been inserted. So, again, a new question for you all. Golden rice was genetically modified to produce more vitamin A. Which part of the plant is modified to achieve this? Again, it's your platform. Go on and show off your genius. Wow, I'm getting a lot of answers, and most of it is the correct. Yes, the answer is option A. Gene coding for vitamin A pathway is added to rice endosperm. We can move forward. Now, exploring synthetic biology. So, we discussed genetic engineering previously. Genetic engineering is a part of synthetic biology. Synthetic biology is overall the union of biology and engineering. Gene editing techniques like CRISPR allow for precise modification in organisms. Synthetic biology designs microorganisms to produce biofuels and pharmaceuticals efficiently. The development of biological circuits enables programmed cellular responses for various applications. Applications in healthcare include customized therapy and production of essential biomolecules like insulin. So, we all realize that synthetic biology is playing a very major role in our lives today. And the final question for today: a student is testing if light color affects plant growth. She uses red light for one group, green light for another, no light for the third. What is the control group in the setup? I repeat, the question is, a student is testing if light color affects plant growth. She uses red light for one group, green light for another, no light for the third. What is the control group in this setup? Is it the red light, or is it the green light, or all the three are test groups, or no light? All right, there is a confusion here. I'm getting a mixed set of answers. So the right answer here is no light. Now, some of you asked what is a control group? So control group is basically the experimental group which does not involve independent variable modifications. You will get to know more about this in the notes. If you go through it, you will understand it better. So, it's time for some random questions. Oh, this one is done. I hope now you know the answer to this question, and you will also go through the notes to identify the reason behind the answer. Now, students, you're free to post your queries on the live chat, and we are here for a few more minutes to discuss them and help you get prepped up completely for tomorrow's quiz.
Hi Shreya, that was wonderful. Maybe we can ask the students to go through their notes once again right now, and maybe if they have any questions, they can go to the live chat and ask us. So, kids, if you guys have any questions, put it on the live chat. Okay, so I can see someone asking as to what is the syllabus. Kids, so this was the entire syllabus that was covered by my entire team. So it is also posted in the round two group if you guys are there in that WhatsApp group. So all the best for that examination. The syllabus, the instruction manual, everything has been pasted.
I see a question for asking for what is plasmid. So, like I explained before, plasmid is the extra chromosomal genetic material that is present in several bacteria and microorganisms. So, we all know that DNA is our genetic information that is stored in our cells. The DNA that we look, the double stranded figure that you may have seen, it is present in the chromosome. Plasmid, on the other hand, is a circular double stranded DNA which is present outside the chromosome within the cell. This plasmid is used in genetic engineering so that we get our modified organisms which have better, which are more which perform better and are more efficient.
Okay, guys. So maybe we can shift our questionnaire to the WhatsApp group that has been dedicated to you guys. So now you can keep commenting on this live chat out here. But now we will be ending the meet. So it was wonderful, guys. That's a wrap. BioQuest champions. We hope this crash course gave you the clarity, the confidence, and the edge you needed for tomorrow's exam. Remember, it's not just about hard work, it's about smart prep, and you've already taken a big step towards it. Whether you joined us for the tips, the practice, or just for the vibes, thank you for being part of this amazing learning journey. On behalf of the whole team, we wish you all the very best for the round two. Stay calm, stay focused, and go ace it. Until next time, signing off with the knowledge and good vibes. Bye bye, guys.
Bye, guys. Hope to see you soon at the IIT Bombay campus.